Hello students, I am your educator Tanvi. Hope you are doing well. Here we are discussing today the second chapter of biology, that is sexual reproduction in flowering plants. So already we have started with unit 1, that is reproduction, and there are 4 chapters which we are going to cover in this unit 1. Firstly, we have already covered chapter 1, that is reproduction in organisms. Today we'll be dealing with chapter 2, that is sexual reproduction in flowering plants, and later on we'll be talking about human reproduction and reproductive health. This will cover our unit 1 of reproduction, and this unit carries 14 marks in your CBSE exam. So let's get started with chapter 2. So to begin with, all the flowering plants, they show sexual reproduction, means all the plants which produce flowers are showing sexual reproduction. So flowers are, you can say, the reproductive organs for the angiosperms. What are angiosperms? Angiosperms are flowering plants. All right. So what are the end products of sexual reproduction? Means what is formed at the end? Seeds and fruits. And flowers, they are the morphological, means they are like the visible appearance is very beautiful, and embryological marvels. And why embryological? Because here the reproduction is going to take place, and embryo is also going to form. We learn about it in detail in this chapter. So this is a longitudinal section of a flower which is taken from your NCERT book itself. Longitudinal means you have cut it from the longitudinal main center axis. The flower, if you see, you can clearly see that there are petals, okay, like rose petals, those are red in color, right? And this one is yellow in color. Then you have these long thin stalk-like structures which are called as filament, and in the you can say at the terminal, this filament is having a lobe-like structure. 2 lobes are there, which is called as anther. Then can you see stigma over here? Okay, this is the place which is very much sticky in nature. And then you can see again a long structure which is called as style, and here you can see it is the ovary. All right, then you can see the green small leaves which are present in flower, those are called as sepals. Ovules are produced inside the ovary only, these are female gametes. And in the flower you have nectiferous area, means nectar, okay, which the bees they suck and produce the honey. So now do you remember that in the previous chapter when we discussed about sexual reproduction, there were different stages we talked about. One was pre-fertilization stage, then fertilization, and post-fertilization. Similarly, when we are talking about flowering plants, we'll be dealing with pre-fertilization events, fertilization event, and post-fertilization event. So let us look at pre-fertilization event first. Okay, what is happening before the fusion of male and female gamete? So few terms which you should remember in this chapter. One is primordium. Primordium means anything which is the basis of something to develop. So it is organ or tissue in its very early stage of development. Like flower bud is there, that is a floral primordium. Okay, so several hormonal and structural changes are going to happen in this primordium so that the flower is ready for reproduction. Okay, so floral primordium would be the flower bud. So later on, when hormones and different structural changes are taking place, then proper flower is being formed. So when flower, totally you can see the petals, you can see the anthers like you saw in the previous slide, that full developed flower is called as an inflorescence. Clear? So primordium is, a floral primordium is floral bud, which will give rise to the whole complete organ, which is flower. And the complete developed flower is referred to as an inflorescence. Now these terms again should be clear to you. The male reproductive structure overall is called as androecium, And the female reproductive structure of a flower is called as a gynoecium. Next, coming to stamen. So stamen are the male reproductive part of a flower. So you can see, we had seen in the longitudinal section of the flower a very long filament and a lobe structure over the terminal end, right? So this whole thing is referred to as a stamen. So it consists of a filament which is long and slender. Slender means thin. So it is a thin stalk. And at the terminus means at the end, it is having a bilobed structure, two lobes like this. Okay, so two lobes are there which are called as anther. Now listen to this term very carefully. Proximal end. Proximal end of filament is this end we are talking about. Proximal end of filament is attached to thalamus or petal of the flower. So what is the petal? You know, the pankhuriyan, like you say, okay, those are petals, and thalamus is the part where the sepals start. Okay, like if you see in this diagram, this is thalamus we are talking about. Okay, so this anther and filament means the stamen, either it is attached to the thalamus or the petal of the flower. And proximal always means near end, and distal, there are two terms, proximal and distal. Proximal means near to something, and distal, from the word distant, means away. Okay? So proximal end of filament means this end, and distal end means this end. Okay, so the number and the length of the stamen, it varies from different variety of flowers. Okay, in different varieties there are different numbers and different length of stamen. It is not fixed. Okay, so here in the other diagram you can see a proper section of anther, which we will discuss in the next slide. So anther is a bilobed structure which is having two theca. Theca are the protective layers. Okay, so you can see that over here, this is a section of anther. If you cut it longitudinally again, you will be able to see these two lobes. Okay, and these are the protective covering in the anther. So two protective layers are there. That is why it is called dithecus. And there is a longitudinal groove. This groove, this is separating the two theca. Okay. Then transverse section. Transverse section is this. Means if you cut anything, like suppose I have this structure, this long structure. Okay. First let us clear. This structure is there. Let me draw over here also. This, if we cut through this axis, this is longitudinal axis. And if we cut like this, it is transverse section. So if we see transverse section, it is a tetragonal structure. Tetragonal means you can see over here, 1, 2, 3, 4. 4 parts are there. So in these 4 parts, what are present? 4 microsporangia. These are structures which will further develop into pollen sacs. And pollen sacs, what they are going to have? They are going to have pollen grains. Okay, so just do it again. Anthers, these are bilobed structure. They are having two protective layers. That is why it is called dithecus. And if you see that the transverse section, you can see 4 parts of anther. Okay, and those 4 parts like this. Okay, 1, 2, 3, 4, like this you can see. So these 4 parts, these are called as microsporangia. It will further, when it will mature, it will form a pollen sac, and these pollen sac are going to contain pollen grains. Now this is the season of pollen grains actually. Okay, pollen grains are nothing but yellowish powdery substances, and these will further act as the male gamete of the flower. All right, so it is very simple. Anther is having 2 lobes, it is having 2 theca, and 4 microsporangia. The structures which are there, they are small in size, that is why micro, and they are sac-like structures, that is why sporangia, which will further develop into pollen sacs. Why pollen sacs? Because they are carriers of the pollen grains. Next, let us look into the structure of microsporangium as it was discussed previously. See, this is the structure of a microsporangium. So this is one microsporangium, second, third and fourth. Alright, so let us look at the layers first of all. So if you see, there is epidermis, which is the outermost layer, endothecium, which is inner to epidermis. Epi means outer, endo means inner. So epi is the outermost layer, endothecium is inner to the epidermis. Then there is a middle layer, you can see over here, middle layer is there, and then tapetum. Tapetum is the innermost layer. Okay, so you can say the outer three layers, which is epidermis, endothecium and middle layer, these are protective in nature and they help in dehiscence. Dehiscence means splitting of anther to release the pollen, because pollen is going to take part in sexual reproduction. The innermost layer is tapetum. What tapetum does? It provides nourishment to these pollen grains, okay, because further they have to take part in the sexual reproduction. And this tapetum, the cells which are there in tapetum, these are having dense cytoplasm. Dense cytoplasm means a high rate of metabolic activities are being carried out inside it, and generally it has more than one nucleus because it has to prepare the pollen grains for sexual reproduction. So it is providing it, you can say, all the metabolic facilities. All right. Next, there is a process which is called as microsporogenesis. So if we split this term, you can see micro, small, spores, genesis means formation. So before discussing what exactly is the formation of these small spores, let us look at sporogenous tissue. So what is a sporogenous tissue? It is a loose type of cells, collection of cells which are present at the center of each microsporangium. So this is microsporangium. This is another microsporangium. So there are 4 microsporangium. This is clear by now. So in the center you can see some cluster of cells. This is called as sporogenous tissue. All right. So now this sporogenous tissue, it will undergo meiosis and it is going to form microspore tetrads. Tetrads means 4. So what is happening? The cells of sporogenous tissue, they are 2n. That is the ploidy, which we discussed in the last chapter also, means they have a complete set of chromosome. Then they will undergo meiosis and they will form 4 daughter cells which are microspore tetrads, which are having half the number of chromosome. That is why n. This is very important. So the formation of microspores from pollen mother cell. Now what is this pollen mother cell? This is nothing but this sporogenous tissue only. So PMC you should remember, this is pollen mother cell. So sporogenous tissue, the cells of this tissue are acting as pollen mother cell. Why? Because they are helping further in producing pollen. So firstly what is formed? Microspores. From where? From pollen mother cell. And this process of formation of microspores from pollen mother cell is called as microsporogenesis, formation of microspores. Okay. And these, why it is called microspore tetrad again? Tetrad means 4 because they are arranged in a cluster of 4 cells like this. Then what will happen? Anthers are going to mature and these microspores, they will dissociate and they will form pollen grains, and thousands of pollen grains are formed. All right. Now let us look at what exactly is a pollen grain. Pollen grain represents a male gametophyte. Okay, means it is the male reproductive structure, and it is yellowish powder, and the variety of architecture is amazing. You can see in this picture that variety of pollen, the different size, shape, color and design. All right, and this is due to the presence of an external cell wall. Generally it is spherical in shape and diameter varies from 25 to 50 micrometers, and it has a 2-layered wall. The outer layer is hard, called as exine, and inner layer is not that hard, it is loose type of cells are present and it is called as intine. The outer layer is made up of a chemical compound which is called as sporopollenin. We will study about it. And the inner layer is made up of cellulose and pectin. These are nothing but polymers of sugars. Let us see what exactly a sporopollenin is. Sporopollenin, children, it is one of the most resistant organic material. And it can withstand high temperatures, very strong acids and alkali. It is very difficult to degrade sporopollenin. So if it is very difficult to degrade sporopollenin, what it means? That it is very difficult to degrade a pollen grain. That is why pollen grain can be preserved as fossils, just because of presence of sporopollenin in the outer layer. Now in the pollen grain, like we have discussed, there is exine and intine. So exine is the outer layer and intine is the inner layer. So exine, in some portions, the sporopollenin is absent. So those portions where sporopollenin is absent in exine, the outer layer, those are called as germ pores. It's just for the transfer of exchange of materials, nutrients. Now let us study about the structure of pollen grain. Now you can see in this diagram, the pollen grain contains two cells. One is vegetative cell and another is generative cell. So obviously it is having two nucleus. You can see one is vegetative nucleus and another is generative nucleus. So let us compare these two cells first. Vegetative cells, these are bigger in size as compared to generative cell, and they have abundant amount of food reserve. It is generally large in shape than the generative cell. Now generative cell, generally it floats. You can see spindle shaped. This is spindle shape, okay? And it is floating in the cytoplasm of a vegetative cell. And it too also has dense cytoplasm and nucleus. You can see over here, this is the exine, intine, germ pore, which we talked about, where sporopollenin is absent. And there are two types of, you can say, two-celled stage. This is the two-celled stage of pollen grain, where there is vegetative nucleus and a generative nucleus. And in 60% of flowering plants, pollen grains are shed at this two-cell stage. Means the pollen grains are going to fall off from the plant at this stage. But in rest of the species, there is a three-celled stage. After forming three cells, this pollen grain is going to shed. So how three cells are going to form? The generative cell, they are going to further divide into two cells. And this is how there would be two small generative cells and one vegetative cells. So some of the pollen grains, they will mature or they will fall off from the flowering plant after this three-celled stage. Now lastly, last part of the chapter of this today's discussion is that whether pollen grains are harmful or they are useful. See, this is the season of pollen grains, I told you, especially those people who are suffering from asthma. It is very difficult for them in these situations because this pollen can cause allergies and it can lead to chronic respiratory disorders. And generally, pollen allergy, it started in India with the imported wheat. The wheat, along with it, carried some parthenium. Parthenium is carrot grass, means that that was having pollen in it. So pollen allergy can be caused in some people, and using different medicines it can be cured, but still the person has to suffer a lot in this type of environment. And pollen grains, what is the useful part about it? They are rich in nutrients. And pollen tablets, generally in abroad, these are available and they can be used as food supplements because they are very rich in nutrients. And it has also been seen that those people who take these food supplements, it increases their stamina. So it has been seen in athletes, their performance, and in race horses, their performance is enhanced by using such food supplements. Now, what is the viability of pollen grain? So viability means that till what time this pollen grain is means viable. Viable means living. Okay, see, pollen grains, they are falling off from the plant. Okay, so till what time it will be in the living form? So it is depending on the temperature and humidity. Like in rice and wheat, after 30 minutes, the pollen grain will lose its viability. Like there is a word, na, validity, that validity has expired now. Like suppose mobile, you have to recharge. Okay, validity is expired means again you have to recharge your mobile. So that is the same thing with pollen grains. Means if till 30 minutes it is used for sexual reproduction, it is fine. Otherwise, again, when new pollen grains will be formed, then it can take part in sexual reproduction. In members of Rosaceae, like rose family, Leguminosae, that is legumes, the pulses, and Solanaceae, like potato, brinjal, they have 1 month viability. Okay, so that is why pollen banks are there. What are pollen banks? Where we store these pollen grains for years. Okay, for experiments generally, because if this like experiments have to be performed in lab, we need pollen grains. If they are not available during one season, what we are going to do, we'll just store them in liquid nitrogen. It is this temperature, minus 196 degrees Celsius, means we are going to freeze them so that whenever they are needed, we can use those pollen grains. All right, now in this lesson today, what we have discussed mainly about the male reproductive part, which is stamen. We studied about its structure. We studied about the anther in detail, what is microsporogenesis, and how a pollen grain looks like, what are its advantages, and what is the viability. In the next lesson, we are going to cover the female reproductive part of plants, and we are going to look into the process of pollination.